Hi, so I will be presenting today. My talk's title is this on? Right, so I'm going to start with the case that that led me to choose this topic. So it's Mr. CJ, a 68-year-old diabetic patient, presented to GSH with a DKA, very severe, pH less than 7. It required significant amounts of inotropes, which was complicated with ischemic colitis as a result. In ICU, the patient developed a candidemia, was put on treatment with microfungin, subsequently a subsequent blood culture, and that was taken after initiation of microfungin treatment, flagged positive after 76 hours, but unfortunately no bacteria were observed. So then a third blood culture flagged positive after 92 hours. On this one, however, initially had no growth. After 2 days, we saw growth on only the plates that had been incubated microaerobically. So I don't know if the ID room has any ideas of what possible organisms we're dealing with, what possible organism we're dealing with here.
All right.
So thanks, Ruan. It sounds like it could be one of the Campylobacter species. I guess Jejana would be the most common.
Okay. Well, great. That's what we got. So I'll be talking a little bit about Campylobacter in the South African setting, and especially focusing on cases of bacteremia. But yeah, that's what this patient had: 2 blood cultures that we isolated a Campylobacter jejuni. So just in terms of a little bit of background on the organism, the Campylobacter species are curved, spiral, motile organisms that are microaerophilic, so they don't like too much oxygen, and they're capnophilic, so they like to grow in CO2-enriched environments. They're commensals of the GI tract of wild and domesticated animals who generally get it early and are lifelong carriers. Relevant virulence factors are Campylobacter jejuni has hypervariable sequences, especially in the areas of the surface accessible carbohydrates. So it has a very variable surface, which has implications for immune evasion. And there are also various toxins that it produces, as well as the ability to evade TLR5, which is an innate immune activation through altered flagellin protein that it has. It also doesn't stimulate TLR9, which is a further innate immune activator because of its rich AT-rich genome. And it is also competent. It can take up DNA from the environment, obviously with implications for resistance, but it tends to like DNA from Campy jejuni, likes to take DNA from other Campy species or other Campy jejuni rather than other organisms. So there's significant diversity. It's not just Campylobacter jejuni. On the left, you can see the significant list of other species which have had case reports and small case series with various associations, notably Campylobacter fetus, subspecies fetus, which is often associated with bacteremia, and then a whole list of others which have specific associations. For example, Campylobacter which is associated with gastroenteritis specifically, which is often reported in dogs. And then on the right, you can see a graph. This is a paper that was published in 2005, reviewing all the isolated Campylobacter species that were isolated at Red Cross Children's Hospital between 1990 and 2005. So you can see Campy jejuni was the most common, but certainly all the rest were also isolated. And why we possibly don't isolate all of them all the time, I'll address in a few slides. So in terms of epidemiology and transmission, it's worldwide, generally contaminated food, water, and unpasteurized milk. In a study in 2004 in Gauteng found 32% of frozen chicken carcasses were contaminated by Campylobacter, and that was culture-based detection. In developed countries where waterborne infections are less likely, the animals tend to be the primary source. You can get spread from direct contact with animals, and nosocomial outbreaks have been reported, specifically neonatal meningitis with Campylobacter fetus. Then there is also Campylobacter species excretion for up to 12 to 40 weeks after infection, and fecal oral person-to-person spread is described, but it's not the most common mechanism. And an uncommon means of transmission may be asymptomatic individuals, though asymptomatic infection is frequently detected. In terms of disease, you pretty much have two pathologies. You have the acute gastroenteritis, which is associated with abdominal pain, fever, and diarrhea with a very low infectious dose, and then extraintestinal findings with bacteremia, prosthetic joint infections, endovascular infections. And these extraintestinal findings are prototypically associated with Campylobacter fetus, but possibly because it possesses an S layer which blocks C3b binding, the implication being it's resistant to human serum. Then incubation period, 2 to 5 days, symptoms generally self-limited, and children under 5 tend to be the most affected group for, in terms of gastrointestinal disease, with a second peak between 15 and 29. On the right, you can see a graph. This is from a large study, the Global Enteric Multi-Center study looking at infants in developing countries, specifically diarrhea. So in 0 to 11 months, it's sitting at the 7th most common cause. 12 to 23, it doesn't feature, but in 24 to 59 months, it's the 3rd most common cause of diarrhea. Now, the important discussion here, bacteremia. Who gets bacteremia? What organism causes it? So in this study from France, which looked at 178 cases of bacteremia, Campylobacter fetus was an important cause, but other Campylobacter species also featured. Now, the critical risk factors that I wanted to point out on the graph is underlying malignancy, liver disease, and medical devices. If you look at the fetus column, 33% of those patients had an underlying malignancy, 34% had liver disease, and 29% had medical devices. And age was also a major factor, with 29% being older than the age of 80. Also note that 41% of those patients with Campylobacter fetus had only fever and no other clinical signs, with only 27% having diarrhea. And Campylobacter infection with other Campylobacter species, while malignancy again was important, liver disease was important, medical devices not as much, and they much more frequently had diarrhea or digestive infections. Positive stool culture, about 1/3 of other Campylobacter infections, but only about 8% of Campylobacter fetus. And that might be because of the isolation techniques, which I'll just address in a second. So in terms of non-Campylobacter fetus, there are the cases where you have a bacteremia from jejuni. That may be due to 3 factors. It may be a transient bacteremia, or a deep focus of infection, often these isolates being also serum resistant, or in an immunocompromised host. And it's very well described, immunoglobulin deficiencies as an important, as a specific immunodeficiency that's important. And the data from that same Red Cross study, Campylobacter jejuni was the most common species isolated from blood, but quite a lot of other species were also isolated. And then we shouldn't forget the post-infectious complications, where obviously the well-known one is Guillain-Barré, where where in serological studies they've shown that 20 to 40% of patients with Guillain-Barré had a Campylobacter infection in the preceding 2 to 3 weeks, but the absolute risk is still quite low at less than 1 in 1,000 infections. But there are also a lot of other post-infectious complications such as erythema nodosum, glomerulonephritis, which have been associated. So looking at isolation, how do we isolate it in the laboratory? Now, if we're talking about stool, there are 3 important aspects to its isolation. Firstly, excluding normal flora, so that can be through selective media or filtration. Then appropriately culturing it, so it needs enriched CO2, low O2, and it takes a while to grow. So you may need 2 to 6 days of culture, and then the appropriate temperature. And then obviously identification based on morphology and gram, as well as biochemical tests. So the media that we generally use to isolate it generally has 3 components: animal tissue digest to provide the necessary nutrients, oxygen scavengers to increase its aerotolerance, generally has a blood or charcoal base, and then antimicrobials are added to select out the Campylobacter and prevent the growth of the commensal organisms, such as vancomycin, amphotericin B, etc. Now, the Cape Town protocol is also a described isolation method where instead of blood agar media, filtration is used because it is quite a small organism. The benefit here is that some of the non-jejunine, non-coli species may be susceptible to some of the media, to some of the antibiotics used in selective media. Additionally, why we possibly don't isolate species other than Campylobacter jejuni or coli on our stool cultures is we don't use incubation in hydrogen, and we keep our plates for only 2 days while some require longer growth, and only really coli and jejuni are thermotolerant, growth at 42, which is what we grow our stool plates at, while we are more likely to detect other species in blood where we incubate the isolates at 37 degrees. So non-culture based methods may have benefits, but they're not necessarily better than culture, specifically because detecting DNA for Campylobacter does not necessarily mean disease. In a study where they looked at 432 diarrheal and matched control samples and did PCR, they found no association in Bangladesh and Tanzania between PCR positivity and diarrhea, in spite of control for cycle threshold value. But they did find an association in Peru. And that's possibly due to the known declining attack rate of Campylobacter over time, and possibly this indicates that natural immunity develops over time, but also develops at different ages depending on the setting. And then enzyme immunoassays, they generally have a sensitivity that's comparable to culture, but they have an issue with false positivity. Possibly, or well, what is theorized and what is described is that they cross-react with some of the other Campylobacter species as opposed to just being specific to jejuni or coli, and those species, the association with diarrhea isn't as clear-cut, so it becomes somewhat difficult to interpret. In terms of treatment, so there was a meta-analysis that looked at treatment and the effect, and this found that that treatment had about a 1.32 day reduction in duration of diarrhea, but also that therapeutic timing was quite important, where in starting therapy earlier had a larger reduction in number of days of diarrhea versus starting therapy late. And then in terms of choice of antimicrobials, which is that I'll be talking about in the rest of my talk, is what do we choose? So the IDSA guidelines advises azithromycin as a first choice and ciprofloxacin as a second choice. And this would be on top of initial empiric therapy, or this would be modified after initial empiric therapy based on risk factors and travel history, where generally they advise treatment in less than 3 months old, immunocompromised with severe disease, ill immunocompetent with severe disease, or international travel with a fever and signs of sepsis, with then modification if Campylobacter is isolated. So why choose the azithromycin over ciprofloxacin, for example? Well, just to start, I mean, the resistant mechanisms that are present and that have been detected, firstly, in terms of aminoglycosides, predominantly the antibiotic modifying enzymes, tetracyclines, the TET-O gene, quinolones, gyre-A gene, beta-lactams, which I'll talk about in the next slide, and then macrolides, the 23S RNA gene A2075G is the most common mechanism, but we'll talk about the gene, which is an emerging mechanism of resistance. So firstly, beta-lactams, and let's look at why this is important. So I've highlighted here piperacillin and cefotaxime, which would be commonly used agents, empiric agents in individuals with severe disease or admitted for presumed bacterial infection. And piperacillin actually, in terms of 100 isolates that were assessed, 0 were sensitive, with 77% being resistant and 23 being intermediate. And cefotaxime, not the best susceptibility profile. This was predominantly in jejuni. In previous studies, it was shown it's predominantly due to penicillin binding proteins, but also due to porins which allow certain antibiotics to enter, but not others. For example, piperacillin has a large side chain which somewhat prevents its effective entry into the bacteria. And due to this, it's primarily why imipenem, cefepime, and amoxicillin are the preferred beta-lactams when treating Campylobacter. Beta-lactamase production is also quite common, but in studies where its detection versus no detection didn't seem to have much of an effect in terms of susceptibility. So it doesn't seem to be an important mechanism in spite of being detected quite frequently. Then macrolide resistance. So the substitution in domain 5 of the 23S RNA gene is the most commonly isolated resistance mechanism. But in spite of this being isolated commonly, why are macrolides still advised as the primary or the most important mechanism, preferred empiric choice for Campylobacter? Well, it may be that this mutation induces a fitness cost in Campylobacter jejuni, especially in colonizing chickens. And this may explain the low prevalence of macrolide resistance, because this food-based spread is the most common and most important means of transmission. And this theory is supported by the fact that there's no such fitness cost seen with Campylobacter coli, and the resistance rates are higher for this species. However, there are strain-dependent compensatory mechanisms that have been identified, and that's why it's important in terms of important for surveillance sites to do whole genome sequencing, in addition to just detecting these specific mutations, to get an idea of emerging, to get an early indication of emerging resistant strains. And then there's also a baseline level of innate resistance associated with macrolides based on an efflux pump. So the gene, the emerging resistance mechanism for macrolides, which is quite important, I'll talk about now. So in 2014, this gene, a ribosomal methylase encoded by B, was first reported in China, associated with Campylobacter coli. So it's as expected, it decreases the macrolide binding by demethylating a single adenine in the 23S RNA gene. And it seems to be mostly a constitutively expressed gene because of mutations in the regulatory region of B, but a small number of isolates have also been found to be inducible. But the real clinically relevant issue with this gene is that it's often harbored in multi-drug resistance genomic islands, which also carries resistance to fluoroquinolones as well as aminoglycosides, making it a real issue if it emerges or if it spreads further. And at least in China, if we look at the figure at the graph on the left, you can see that in Campylobacter coli, it really has had a year-on-year increase in prevalence, and then now it has also started to be isolated in Campylobacter jejuni, which is obviously a much more common cause of infectious diarrhea. The prevalence is still quite low, and it's largely confined to Campylobacter coli in China, but it can be transferred by natural transformation, and it has actually recently been identified outside of China, specifically in Spain. And again to ciprofloxacin, but also surprisingly higher resistance for erythromycin and azithromycin, and I'm uncertain as to why that would be. What is quite interesting is that a paper looking at azithromycin compared to erythromycin resistance in the USA found that there tended to be correlation as opposed to what we're finding here in South Africa. And in the one that they did find discordance, there was a specific ribosomal protein amino acid substitution. And it would be quite interesting to look at the South African isolates to see if there was a similar finding. But obviously, I couldn't find any whole genome sequencing data on Campylobacter jejuni and coli. So the last two slides, I just want to show results from two other papers, which I found somewhat difficult to interpret and somewhat concerning if the findings are an accurate representation. So this is data from a study done in the Eastern Cape, which looked at environmental isolates from milk and from water. Again, Campylobacter jejuni most isolated, but what was really interesting was the level of resistance in these environmental isolates, with a proportion of azithromycin and erythromycin being up to 40% resistant, only really imipenem being largely susceptible, susceptible across the board, both for Campylobacter, other Campylobacter species and Campylobacter jejuni. And then a meta-analysis and systematic review of stool samples in sub-Saharan Africa, with the majority coming from East Africa, again found quite a lot of resistance for erythromycin, and less so for ciprofloxacin, and also quite high resistance for gentamicin. So I looked into this data specifically, but unfortunately they didn't mention in this study specifically which criteria they used for determining resistance. But most of these isolates were Campylobacter jejuni. So yeah, I found that quite interesting. And yeah, that's pretty much what I wanted to show, just a broad overview of Campylobacter and the susceptibility and resistance profiles across South Africa, as well as in the rest of sub-Saharan Africa. Any questions?
Thanks very much, Ruan. Very nice, as you say, very nice overview. While people are thinking of their questions, and if you do have questions, if you could pop your video on, that would be great. I'm glad you sort of brought up at least the environmental side, because I'm sure this is very much one of those bacteria that are important in relation to One Health. So in the animal side as well, your reference to 30% of chickens in KZN being infected will worry our colleague from KZN who's on the line, Eunice. Is that an old study or a new study? And what is known about Campylobacter in the food production animals in South Africa?
So that study, if I just check my references, that study was from 2004. So other than that specifically, I'm not really aware. In addition to the frequent finding, I mean, it was found in about 32%, but Salmonella and Shigella were also found in a not insignificant amount. I think both just under 20%, if I remember correctly. And then the latest study from the Eastern Cape, that one from environmental sources, that's quite recent. That was published in 2020. So I mean, my worry or concern, especially at least for the East African data, is that with this high amount of erythromycin resistance, in addition to gentamicin resistance, is it not a hidden gene that is just not being detected, considering it spreads with a multi-drug resistant island? But then the ciprofloxacin susceptibility was better there. So I guess theoretically that against that.
And the whole question of mass drug administration with azithromycin to reduce child mortality, so mass use in drug administration in children would be an interesting intervention, well, worrying intervention. We have a hand up from the seminar room.
Hi, Ruan. Thanks for a great talk, it's Sean. So could I ask you, I mean, you showed a lot of data around susceptibility for different species, different antibiotics, different settings. Could I ask you just to kind of summarize it for the clinicians? I mean, what's the kind of take-home message? What I got from what you showed, from at least what was most likely to be clinical isolates in Cape Town and in South Africa, it looked like, mainly in Cape Town, it looked like erythromycin had a higher proportion were susceptible to that antibiotic compared to azithromycin. And we honestly prefer not to use erythromycin in general because of its poor tolerability and QT issues. So did I interpret that correctly? And just can you give us just a high-level summary of what you think we should be doing in light of the data that you've shown? And then also just a second quick question, if there's time. What are the treatment recommendations for invasive infections? So it's with Campylobacter fetus, which cause bloodstream infection, which is a more severe illness, which the treatment duration, maybe choice is different compared to just an acute diarrheal illness. Thank you.
Okay, so if we split it into the Campylobacteriosis gastroenteritis and the invasive infection. So based on the NICD data as well as the GSH data, with Campylobacter jejuni, at least from enteric sources being the most commonly isolated one, it does seem that our macrolide resistance profile, it's macrolide resistance in South Africa at least, is still infrequent. So I think if we go over the 2017 IDSA guidelines, I think that treatment recommendations hold. Azithromycin or erythromycin arguably would be the drug of choice for Campylobacter. And looking at the NICD data for both erythro and azithromycin, the susceptibility is high. So there's not much divergence, and this is at least for public healthcare samples. That one study from private healthcare that was somewhat different, but that was one sample from one site and one lab. So I wouldn't necessarily go broadly based on that study. But from the large number of samples and from our side as well, I think azithromycin as a first choice for gastroenteritis for Campylobacter would be a reasonable choice based on available data. Then for invasive infections, I couldn't find a specific guideline, but based on the study from France, which showed that lack of treatment with an effective antibiotic so we had an 88% mortality versus 15% with treatment with an effective antibiotic. I think if we want to ensure that we are treating with something that's effective, regardless of the specific species, where we can see that, for example, from our GSH data, we saw mostly Campylobacter, but also coli as well as other species. I think gentamicin or imipenem would be your preferred agents